This is the first lecture of the course. So it is chapter 1 and it talks about the microbial world and you. This chapter is just an overview of what are microorganisms and how do they affect us in positive and negative ways. What are some of these diseases they cause? And it also talks about some important dates, important people that have had made major contributions in microbiology. Now, what is the definition of a microorganism? So the name describes exactly what they are. So these are organisms that are too small to be seen with the unaided eye, meaning that you need some type of microscope to be able to view them. For example, bacteria. Okay, when we think of a bacteria, we think that it's something small and we cannot see it with the unaided eye. For example, if you take a look on your palm, the palm of your hand, there is bacteria there, but you cannot really see it. You just see skin, you see, maybe other things, but you are not going to be able to see bacteria or any microorganisms. So micro represents something that is small, microscopic, and an organism is considered something to be alive. Now, in this course, we are going to focus on several important microorganisms, which are bacteria, fungi, protozoa, microscopic algae, viruses, and also hamlets, which are parasitic worms. Now, the reason why we do study them in this course is because their infective form is going to be the eggs, and we cannot view the eggs with the unaided eye. We need a microscope to view them. So we are going to include them in this course. Now here, if you see this image, it just gives you a diagram so you can compare, or you have an idea, I guess you can compare the sizes of microorganisms compared to other biological structures. So first we have the first unit, which is atoms, which is about like 0.2 nanometers, which is very, very small. You actually need an electron or even an atomic microscope to be able to view them. And then, of course, atoms make small molecules, and then small molecules start making what you find in the cells. For example, lipids are found in the plasma membrane. Proteins are going to be made of amino acids, and then you have ribosomes, which make proteins in the cells. And then you start to see one of the microorganisms, which are viruses. This is one of the forms. There's other structures that make up viruses. But viruses are around a little bit under 100 nanometers. And then you start to see the first, the smallest bacteria, which is chlamydia. You have mitochondria, which is the organelle that is found in cells, and it makes ATP, makes energy for the cell. And if we compare it to most bacteria, they're kind of on the same, size range, okay? And then we have the nucleus, and we have what most animals and plant cells look like. So as you can see, most animal cells are going to be in between 10 micrometers to 100, and bacteria are maybe like 5 micrometers. So as you can see, they're way smaller than our cells, so they can literally go inside of our cells. And these are just the different types of microscope that you can use to view them. Here we have the electron microscope, which uses electron beams to be able to view these structures. We have here the light microscope, which uses light. And with the light microscope, you start to see most bacteria in most of the cell's structures. Next, we have a fish egg, which we could see with the unaided eye. We can't really see its internal features that well, but we could still see it. And then we have the human, which is, a little over a couple of meters high. Okay, so as you can see, compared to our height, compared to the fish egg, something that we can see, we can't see our own cells. We cannot see, of course, all bacteria, viruses, and all these structures here. So that is what this course is going to focus on, meaning microorganisms that we cannot see with the unaided eye. Now, when we think about a microorganism, a microbe, we think that about something bad. We say, they're bad for us. When you think about microbes, you just say, ew, they're just nothing good. So how they affect us in a harmful way or in a negative way is that they're going to be pathogenic, meaning they're going to cause disease not only in humans but in other organisms as well. Now, there is thousands and thousands of microorganisms, but only a few are going to be causing disease. It's just that, of course, disease is bad for us. Nobody likes being sick. Some of the diseases can even lead to death. So when we think about microorganisms, we associate them off the bat that they cause disease, even though only very few out of the thousands and thousands of microorganisms are going to cause disease. And then another harmful way is that they are also spoil our food. I'm not sure if you guys have ever left bread outside or just fruits or vegetables, and you start to see these moldy structures over them. That is microorganisms that are in the air, just pretty much spoiling and damaging your food. So these are the two ways of how they affect us in a negative way or a harmful way. Now, believe it or not, there is way more ways that they're going to affect us in a positive way. Okay, so I know that they cause diseases, they spoil our food, but the other percentage, which is way more microorganisms, they're going to affect us in several, or actually, yeah, several positive ways. And those ways are that they help us decompose organic waste. There is microorganisms, specifically bacteria, that they like to eat the waste and they're going to convert it into something organic that is not going to be toxic to us humans. They also help us generate oxygen by photosynthesis. For example, algae, some type of protist, and also bacteria. They are capable of producing oxygen through photosynthesis, and we know that as humans, we breathe oxygen, so oxygen is very, very important for our survival. So those microorganisms affect us in this positive way. They also help produce chemical products such as ethanol for fuel, acetone, and even vitamins, which we know that vitamins are also essential for our health. They even help us produce foods, specifically fermented foods such as vinegar, cheese, and bread, which most of us include them in our diets. They also help us with the manufacturing of cellulose, which is used for clothing. And they're also going to help us make treatments like insulin, antibiotics, and other specific medications. They're also used for pest control, meaning the insects or the pests that eat the crops. There is some microorganisms that actually are very toxic to those insects, and they're going to prevent them from damaging the crops. And then tying back to the first bullet point here, there is microorganisms that also help us recycle water. And I'll show you towards the end of the lecture how that happens. Okay, so one of the things that we also have to talk about when we talk about microorganisms is the microbiome. Now, the microbiome is composed of microorganisms, and these microorganisms are bacteria, protists, even fungi, and they're going to help maintain our health, meaning they're going to help us fight off other microorganisms. They're going to help us maintain a certain pH. They're just going to be very healthy for us, and they're going to give us or help strengthen our immune system. So normally the adult human averages or has about 30 trillion body cells. Okay, so these body cells, like I said, each one is going to have a specific function in our bodies. But our body also harbors or has 40 trillion bacteria cells. Okay, when you say 40 trillion, you say, hey, we have way more bacteria cells than we have of our own body cells. But remember that bacteria are way smaller than our own cells, so that's why we have 40 trillion of them. Now, they are found on your body, like on your skin, even on your hair. They're just found on your body and also in your body, in your mouth, in your intestines. They're even in your lungs, and they're even in your reproductive organs as well. So these healthy microbes that are going to help us fight other microorganisms, help us maintain a good normal flora, and even train our immune system are going to be called the normal microbiota, meaning they're going to help you maintain good health. Like I mentioned, they're also going to prevent the growth of pathogenic microbes, meaning microbes that are going to cause diseases. They actually fight them off. They prevent them from getting nutrients. They produce toxic materials that are going to be harming those microbes. So they're just going to be helping us fight off microbes. And they're also helping our immune system get trained to discriminate against threats. So meaning, since we do have bacteria living in or on our body, the immune system knows how to determine if it's a bacteria or if it's just our normal cells. So I know it's hard to believe, but no, just know that having microorganisms in and on your body is actually beneficial. Only if they are, of course, composing of the normal microbiota. Okay, so now that I talked a little about microorganisms, why do we want to know a lot about them? Why do we want to have knowledge of these microorganisms? Well, the first thing is, of course, you want to know how to prevent them from spoiling our food. Okay, food is expensive, especially right now. So of course, you want to find ways of how to prevent them from spoiling our food. At what temperature should we cook, our food? At what temperature should we put our food in the fridge? Should we freeze a certain type of food? So just knowing how to prevent food spoilage. And then also a very important one is, of course, we need to know about these microorganisms to prevent disease. What specific ways can we prevent them? Is there a certain medication? Do we need a vaccine? Just how to prevent disease. And then, of course, understanding the causes and transmission of the disease to prevent epidemics, to prevent people from getting sick of these infectious disease caused by a microorganism. Okay, so just like any other living organism, microorganisms are also going to be named and classified, okay? And each organism is going to have two names. They're going to have a genus and they're going to have a species or specific epithet. So just think about the organisms having a first name and a last name, okay? First name and last name are going to be specific to an individual. So here we have the genus, think of it as the first name, and then the species is going to be the last name. Now, whenever you are writing these, scientific names, there is certain rules that have to be followed. The first one is that if you are writing or typing any scientific name of an organism, it has to be either in italics or it has to be underlined. Okay, either or, both are correct. And then the first letter of the genus has to be in capital letters, and the remaining of the genus name and also the species name are going to be in lowercase. All right. So when you look up these names, you're gonna see that they are names that you don't even understand or you find that they're not common. This is because they are in Latin. And the reason why they're in Latin is because they are used worldwide. The names are used the same in every single part of the world. The names could be descriptive, meaning they could describe the shape, the color, of the microorganism, or they could even honor the scientist that discovered that specific microorganism. I do want to say this, for your exam, there will be several names, several examples, and only one of them is going to be correct. So make sure that you know these rules, because there is going to be a question on this for sure on your first exam. All right, so just to give you some examples, remember that the names have a genus name and they have a species name. Both names are in italics, and only the first letter of the genus name is going to be capitalized. So for example, here we have Staphylococcus aureus. Now, Staphylococcus aureus is a very common bacteria that you find on your skin and in other parts of your body, but it's mainly found on your skin. Now, as you can see, the first name that you see here is going to be the genus, okay? And as you can see, the first letter, which is the S, is a capitalized, all right? And then the second name here is going to be the species name, and as you can see, it is going to be all lowercase and of course in italics. So here this name is just describing the bacteria. So here Staphylo represents clusters, and spherical, meaning coccus, is going to be, they have a spherical shape. Okay, so this is what Staphylococcus looks like. As you can see, it's little spheres, okay, and they are all in clusters. So Staphylococcus is just spherical bacteria forming clusters. And then aureus is going to be the color of the bacteria, which is going to be this gold color. So that's what we see here. And then next, very, very common bacteria is Escherichia coli. Once again, the first letter of the genus is in capital letters. Everything else is in italics and in lowercase. Okay, so here Escherichia is actually from the discoverer, meaning the scientist that discovered, which is going to be Theodore Escherich. And then coli, it just represents where the bacteria is found. And Escherichia coli is mainly found in your large intestine or in the colon. And this is how it looks like. Okay, so the scientific names, I know that Staphylococcus aureus and Escherichia coli, they just, sometimes they're even hard to pronounce even for me, and they're very long names. The names can even be abbreviated, and instead of having to write the whole genus name, all you have to do is just assign it its first letter, okay? And then the species name or the epithet is what is written completely. So here we have E. coli, okay? So as you can see, Escherichia is just abbreviated with an E, and then you just have coli. And then as aureus, Staphylococcus is abbreviated with the S, and then you have aureus. By the way, I see here a mistake, okay? So here as aureus is supposed to be in italics, all right? So on your exam, if you see this specific format, that is going to be one of the answer choices that is wrong because it is not in italics, okay? So for your exam, like I mentioned, I'm going to give you several examples. They're going to be, only one is going to be correct, so please make sure you do know the rules for this. There is going to be a question for sure. Okay, and then next up, we're gonna just briefly talk about the different microorganisms. So do not worry if you don't understand something right now. There is going to be a full chapter that is going to talk about each one of them. But just to briefly introduce you, I'm going to talk about them in this lecture. So like I mentioned in the first slide, the microorganisms that we're going to focus on are bacteria, which there is many bacteria. There's many shapes, many colors. But for example, we have here this one that looks like little rods, and they're also forming clusters. We have archaea, which you really never hear about archaea because it really doesn't cause disease, it doesn't harm us or even affect us in a positive way. But just know that archaea are considered a microorganism. We do have fungi. Fungi are considered a microorganism, and you're going to learn that they could help us or they could also affect us in certain ways. Protozoa are single-cell organisms, and they just have many shapes, many sizes, many colors. Algae are going to be photosynthetic organisms. Here are microscopic algae. Viruses are going to also be put in this category, even though they're not considered organisms because they're not considered a living cell. They do affect living organisms, so we're going to include them in this chapter. But for example, here we have a nerve cell, and then we have the Zika virus that is just infecting the cells. And then we also have the multicellular animal parasites. When we think about a parasitic worm, you definitely could see it with the unaided eye. But we are going to include it in this chapter because the infected form, which is the egg, that looks like this, it is microscopic, you cannot see it. And then also they do cause disease as well in humans. So we are going to include them in this chapter or in this course. Okay, so first up we have bacteria. Now bacteria are considered prokaryotes, meaning pre-nucleus, and that means that they do not have a nucleus. Its DNA, its genetic material is not enclosed by a nucleus. They are single cell, meaning that they're only composed of one cell. Every single bacteria is only composed of one cell. For example, here we have just a bunch of bacteria. Each bacteria is only composed of one cell, but they're just going to like to live in little communities or just in little chains or clusters. They do have peptidoglycan cell walls, which is a carbohydrate composition, and you will know exactly how the peptidoglycan wall looks like and how does it function when we get to that chapter. They do divide by binary fission, which is a replication, I guess, a pathway that is not as complicated as mitosis. So for example, here you have your parent bacteria. The circular structure is going to be its chromosome. Here you see that the chromosome is duplicated. You have two copies now, and then you start to see that the cytoplasm and the cell starts to split into two. So at the end, you're going to have two daughter cells that have the exact same genetic information as the parent cell. It is way less complicated than mitosis. They do eat, or they get their nutrition from organic and also inorganic chemicals, and some of them are capable of producing oxygen and sugars through photosynthesis, which is kind of like what plants and algae do. Some of them can swim, and they can have flagella, these little hair-like projections at the end, which are going to help them with movement, swimming, and just any other form of locomotion. Next, we got archaea. And like I mentioned, archaea, you really do not hear a lot about the specific classification of microorganisms. The reason why is because they really don't cause any harm to us humans. They are considered prokaryotes, meaning they do not have a nucleus. They do not have a peptidoglycan cell wall, meaning that they have another carbohydrates composition. And some of them might not even have a cell wall at all. Now, archaea are very well known for living in extreme environments, meaning environments that most of the organisms are not able to survive. For example, extreme holophiles, which are going to be like living in places where the salt concentration is very high, that, for example, a lot of organisms are not able to survive, but they are capable of doing it because they're saline tolerant. Some of them are even extreme thermophiles, meaning that they like to live in high temperatures where not, or at least most organisms can't survive. Some of them are even considered methanogens, which are going to produce methane, which is kind of like a, I guess not, I guess you could say a toxic product that is also used for fuel and other, I guess, products that we use. Like I said, they're not generally not known for causing disease in humans, so that's why you really do not hear about them. But they are still considered a microorganism. Next up, we have fungi. Okay, so now fungi are going to be considered eukaryotes, meaning that they do have a nucleus that is going to surround its DNA or its genetic material. Compared to bacteria, they're going to have chitin cell walls, and they're going to absorb their organic chemicals for energy. It's kind of like eating, but they're just going to absorb it from the environment, and they're going to break those organic chemicals for energy. Now, there's I would say three forms of fungi, which the first one is yeast. Now, yeast are going to be unicellular, meaning composed of only one cell. And yeast are mainly used for like making bread. For like, I'm not sure if you guys have ever try to make bread. You have to add yeast to fluff up the dough. And then you have molds, which are most common known for spoiling food. For example, here we have a piece of bread, and then you have like these weird looking green color. That is considered fungi. And then you have your mushrooms, okay? So molds and mushrooms are multicellular, meaning they're composed of many, many cells. Now, with mushrooms, you do have to be careful because some are very nutritious. Some of them we could eat and they're just they have a lot of vitamins and a lot of nutrients for us. But some of them are even poisonous. Like, for example, this mushroom here, if you do eat it, it can be poisonous. It can even lead to death. And then some of them just make you hallucinate and they're not very nutritious either. Next, we got protozoa. Protozoa are also eukaryotes. That means that they do have a nucleus surrounding their genetic material. Now, they do absorb just like fungi, and they can also ingest organic chemicals. So they do have these little, very tiny mouth parts where they can ingest the food. They can be motile, and they can move through pseudopods, which in the case of our little amoeba here, they're kind of like little arms that are stretching out. This is the food, and they're just going to just surround the food, and then that is going to get engulfed. Some of them even move through cilia, which are these little hairs. I'm not sure if you guys could see them, but it's like tiny, tiny hairs that are going to help them for movement, and then also flagella, which are longer projections, like this one here, and this one here for movement and all of that. The protozoa can be free living, meaning they could just live on their own, mainly in aquatic environments or moist environments. And some of them are even parasitic, meaning that they actually get in our bodies and they start stealing our nutrients and cause diseases that way. They're unique also because they can also perform photosynthesis. Not all of them, but some of them can do photosynthesis, and they can reproduce sexually or asexually as well. Next, we have algae. Algae are also eukaryotes. They do have cellulose cell walls, which very similar like plants. Plants also have cellulose cell walls. They are found in fresh water, salt water, and even the soil. So this tells you that they like to live in very moist, just environments that have enough water for survival. Just like plants, all algae are capable of performing photosynthesis for energy, and they're going to produce oxygen and carbohydrates, okay? And then they could also reproduce sexually, and they could also reproduce asexually. Next, we have the multicellular animal parasite, which are parasitic worms. So they are considered eukaryotes. They do have a nucleus that surrounds their genetic material. They are considered multicellular animals, meaning composed of many, many cells. They are not, like I mentioned, they're not considered microorganisms because you could see them with the unaided eye. But they are going to still be parasitic, meaning cause disease in humans, and they're I guess their egg stage or their larva stage, they're considered microscopic stages. Okay, like here we have the egg. We can't really view it with the unaided eye, so we're gonna include them in this chapter. We have flatworms, which literally they're just flat. For example, this one here looks like just a long piece of tape, which is just flat. And then we have the round worms, which are just round worms that look like this. Like I said, they are very scary because you don't really know, or you can't really see your food is contaminated with the eggs because you can't really see them. And they do travel to pretty much most of the parts that are in your body, like your eyes, your mouth, and then even, for example, we have this ankle. This person, had a worm even, in its ankle, so it's just, if they're not treated on time, they can be very, very scary. And lastly, we have viruses. Now, viruses are not considered a living organism. The reason why is because they do not have the mechanisms to function as a living cell. They are considered acellular, meaning that they have to be inside a host cell for them to replicate and just reproduce, pretty much. They can either have DNA or RNA, and it is protected by a protein coat and even being closed by a lipid envelope. They, like I said, they do replicate, but they can only replicate when they are inside a living host cell, and they can't really replicate when they are outside. So if they're outside and they cannot get inside a host cell, then it just pretty much dies and it doesn't perform its function. We have a virus that looks like this. It has a little head and it has like little feet. This one looks like a little tube, and then this one looks like a little circular virus. So as you can see, they have different shapes. Their goal is to infect a living host cell. Okay, so now going back to the classification of microorganisms. Like I mentioned, each microorganism is going to have a scientific name, and this is just based on how they look or who discovered them, where they're found in the bodies or their habitat. Now, there's so many of them that, of course, we have to classify them and we have to put them in a specific category compared to other organisms. Now, this specific mechanism of classifying microorganisms was developed by Carl Woese in 1978, and he established that all organisms could be put in one of the three domains, okay? And this is based on cellular organization. Either organisms can be a bacteria, they could be archaea, or they could be eukarya. So we already got introduced to bacteria, and we know that bacteria are single-cell organisms that are prokaryotes. We have bacteria that are considered thermophoga, which like to live in very hot environments. We have gram-positive bacteria, which we'll get to that in the next chapter. We will learn what that is exactly. So we have all these different bacteria, and then we have archaea, which is another classification. These are also prokaryotes, but they are the most extreme environment prokaryotes. And then we have eukarya, which is going to be composed of protists, fungi, plants, and animals, like us humans. So as you can see, we have fungi here, amoeba, plants, and they are all considered eukaryotes because they have a nucleus surrounding their genetic material. For your exam, make sure you know the three domains, which are bacteria, archaea, and eukarya. Okay, so now that we establish the different classifications of microorganisms and we more or less know what a microorganism is, now let's go back to important dates and important people that have made a very great contribution to microbiology and biology in general. So back then, people really didn't know much about microorganisms, and the main reason is because you can't really see them with the unaided eye. You know, if you can't see them, how are you going to know they exist? But in 1965, Robert Hooke, he was actually one of the first individuals to be able to view cells. Okay, so he actually used a microscope that looked like this. As you can see, this looks odd. It doesn't really look like the microscopes that we use today. It fits on the palm of your hand, which is very, very small. Here's the lens, and you just put your eye over the lens. This is where you put the specimen, and then you have your specimen positioning crew to move the specimen slide. The focusing control, so the little focus to prevent the image from being blurry, and then you move your stage up and down. So as you could see, it is, like I said, very small. It is nothing compared to the microscopes that we have now. But this little microscope that you see here made a great discovery because Robert Hooke actually described or discovered cells, which he saw little boxes. They were just little boxes, and he said, okay, what are these boxes from? I think it was a blood sample that he was viewing, and he just saw little boxes, little cells. So you could say that he was the first one to discover or view cells. So because of his discovery, it marked the beginning of the cell theory. And the cell theory states that all living things are composed of cells. Okay, so if you're considered something to be alive, you have to be composed of cells or only one cell. Now, because of his contribution, next we have Anton van Leeuwenhoek, and he said, okay, you use a microscope and you were able to view cells. So Anton van Leeuwenhoek, he discovered the first animalcules, or the first single-cell organisms, in 1623 to 1673. So these animalcules are single-cell organisms like protists or bacteria, and he was able to view them also with this specific microscope here. Okay, so I know that it's very hard to confuse them, but Robert Hooke was the first one to discover cells, and Anton van Leeuwenhoek was the first one to discover single-cell organisms, or animalcules like he called it. Okay, so now next up we have Louis Pasteur, which he was very important because he disproved spontaneous generation. Now back then, individuals thought that life just came to be alive, or to just came to be in existence by a vital force. Okay, from non-living matter. So meaning that you probably just have maybe some water and maybe some sun, and then all of a sudden you have a cell, or you have a living organism. Now, Louis Pasteur, he said, okay, wait a minute, I think you're wrong. I think that cells have to come from pre-existing cells. Okay, so life does not happen just spontaneously out of nowhere by a vital force. There has to be an existing living cell for it to duplicate and give rise to a new cell. So he was able to do this experiment, which I'll go to the next slide. And he was able to prove that life comes from pre-existing cells. Okay, so before I go on to that, individuals, they did this experiment where they had this flask that you see here, and this flask had bone broth. Okay, so we know that bone broth comes from an animal, and they said, okay, we're gonna have bone broth. It comes from an animal, okay, and we're going to boil the water, okay, and after boiling the water, we're gonna kill every single organism that is alive. So they tested it, and then they said, okay, there is no living organism. They let it sit for a couple of days, and then they took another sample, and then they said, and then they found out that there was cells that were alive. So they said, see, at the beginning we didn't have any cells, and now we have cells. That lets you know that life or cells come spontaneously through a vital force. But then Louis Pasteur said, hey, wait a minute, did you know that there is microorganisms roaming in the air? What if those microorganisms got in your flask? Okay, so he said, I'm going to do a separate experiment, and he used this swan-shaped flask, or S-shaped flask, which looks like this. And he did the same thing. He put bone broth in the flask, and he boiled it. And then he established that there was no microorganisms. And if there was any microorganisms in the air, they were going to get stuck here. Okay, so he let it sit for a couple of days. He tested the bone broth, and he discovered that there was no microorganisms present. So this major experiment just changed the whole, I guess, theory of, biology or microbiology. And now we can support the theory of biogenesis. So meaning that Pasteur, he demonstrated that microbes are responsible for food spoilage because they are roaming in the air. And he also established that a cell has to come from a pre-existing cell. It does not come just through spontaneous vital forces. It has to come through a pre-existing cell. So here we have the experiment, and there is a museum in Paris, the Pasteur Institute, where they still have this flask. And over 100 years later, they still have not found any contamination or microorganism in that flask. So that tells you, and that still keeps supporting, that cells do come from pre-existing cells. Okay, so next, moving up to history. So Pasteur did show that microbes are responsible for fermentation. So meaning, aside from establishing that, life comes from pre-existing cells and not from non-living matter, he also established fermentation. Now, fermentation is the microbial conversion of sugar to alcohol in the absence of air. So believe it or not, microorganisms are capable of breaking down sugar, and they're able to produce alcohol or sometimes even lactose when there is no oxygen available. Okay, so microbial growth is also responsible for spoilage of food and beverages. So we know that, like I said, microbes are in the air. Microbes are just roaming the air. We cannot see them. Since they're roaming in the air, they are going to spoil your food and your beverages. So that's where he came up with the term pasteurization, where is the application of just applying high heat only for a short amount of time, just enough to kill harmful bacteria, but for it to not damage the specific product. In this case, it will be milk. Pasteurization is still, this method is still used in this industry, where you just apply just very little, I guess, heat for a very short time, and that only kills the harmful bacteria. Okay, and this was again back to the idea that there is microbes present in the air. Next, we get to Joseph Lister. Okay, so Joseph Lister, he changed the whole protocol for surgery, I guess, techniques. In the 1860s, he saw what Louis Pasteur did, and he said, okay, Louis Pasteur is saying, and he did research that there's microbes in the air, and that spoil the food. And he said, okay, what if these microorganisms are also in, surgical rooms or just in the hospital? And that's why our patients are getting even more sick or just more wounded when we're doing surgeries. So he established the use of chemical antiseptic, phenol, which is a type of alcohol, to prevent surgical wound infections. Okay, so he actually did this whole, procedure or protocol where he had to sterilize and had to clean the equipment and even their hands to prevent the microbes from infecting patients that are going to have a surgery. So back then, they really didn't clean the equipment they use or the surgery equipment they use very well, but he was one of the first ones to do it. And after that, the number of people that had wound infections or surgical wound infections decreased significantly. So thanks to Joseph Lister for that. Next, we got Robert Koch. So in 1876, he discovered that a bacterium causes anthrax, which anthrax is a very deadly bacteria. It could affect your skin, it could affect your gastrointestinal tract, and even affect your respiratory system. So because it was very, very harsh on humans, he established, the Koch's postulates, where he demonstrates that a specific microbe causes a specific disease. So meaning a specific disease is only caused by a specific microbe, and nothing more than that. So once again, Robert Koch, he established the experimental steps for directly linking a specific microbe to a specific disease. And by the way, all the names and important people that I mentioned, make sure you know their contribution to microbiology, because there is going to be several questions about this. Okay, so now talking about vaccination, which we know that vaccination is, I guess, a prevention technique that is used to help you be protected against a specific microorganism or disease. So in 1796, Edward Jenner, he was the first person to, I guess, use a vaccination on a person, okay? Now, when we think of a vaccination, we think about an injection, but just know that vaccines can be taken orally. It's not only through a vaccination. Well, Edward Jenner, he, was at the time he was treating people that were sick from smallpox, and people were dying. A lot of people were getting sick from it, specifically children. So he just, was very desperate. He said, I need to find a treatment. I need to find a cure, something. So he was, just there, just thinking of what to do, and he noticed that the milkmaids were not getting sick. Were the milkmaids are the ones that milk the cows, and he noticed that they were not getting sick. And he said, "Okay, that's weird. How come they're the only ones that are not getting sick?" Well, he discovered that the cows that they were milking, they had cowpox, which is the same as smallpox, but it's just for cows. And he noticed that they, were getting the bacteria, the virus in them, but their immune system, was just able to just, protect them from it, and it gave them immunity against smallpox. So this was very important for him. He actually said, okay, we might have something here. So what he did, he went to a cow that had cowpox, and he took a little sample from their scalp. So he took like a sample from them. He opened a cut, so he made a cut on one of the kids. He inputted the cow sample on the cut, and he noticed that the child started getting better. And this is because the cowpox virus was training the immune system against smallpox. So this was just a change for the whole medical field. And this was the first vaccine that was administered. It was actually administered to a young boy that had smallpox. So that's why the word vaccination comes from the word vaca, which is cow in Spanish. And then this is just pretty much giving you protection or giving you immunity against a specific disease. All right, so talking about vaccinations, now we're going to talk about the birth of modern chemotherapy. Now, when we think about chemotherapy, we think about a treatment that is for cancer. But just so you know, a more general term, chemotherapy is going to be any treatment of disease that uses chemicals to treat that disease. So chemo represents chemicals and therapy is treatment. So chemotherapy is the use of chemicals to treat a disease, not only cancer, but just any disease in general. So chemotherapeutic agents, they are used to treat infectious diseases, and they can also be through synthetic drugs, meaning drugs made through like in labs and just man-made drugs, or they could even be used through antibiotics, which is we know that it's a treatment for bacterial infections. Now, antibiotics are chemicals produced by bacteria and even fungi that are going to use to inhibit and kill other microbes. So here's one of the positive ways of how microorganisms, affect us in a positive way. Bacteria and fungi are going to be microorganisms that produce antibiotics, and those antibiotics are going to be used to kill other microbes and prevent a disease or cure a disease. Just some terms, talking more about the modern developments in microbiology. Bacteriology is going to be the study of bacteria. So here the first bacterial means bacteria and ology is the study. Okay, so bacteriology is the study of bacteria. Mycology is the study of fungi. So when you hear the word myco in scientific terms, just know that it's referring to fungi. Okay, I know that it's hard to associate to fungi, but just know that mycology is the study of fungi. Virology is easy to remember because here the first couple of letters represent like a virus. So virology is going to be the study of viruses. Parasitology is going to be the study of protozoa and parasitic worms. And immunology is going to be the study of immunity. Okay, so I think these are very easy to remember because kind of like the first couple of letters kind of give you what the study is about. So she made a high contribution, a great contribution on that for establishing a certain characteristic of a microorganism and saying that it could train the immunity on that specific component. Okay, next we have molecular genetics, which more terms. So molecular genetics is the study of how microbes inherit traits. And we are going to learn how these microbes inherit their traits. They could either do it from the parent cells or they could even do it from the same generation cells. But we'll get to that in the next couple of chapters. Molecular biology is a study of how DNA directs protein synthesis. So the synthesizing of proteins, we know that um, proteins, they come from the the translation of RNA, and RNA comes from the translation of transcribing of DNA. Okay, so we'll learn the flow of genetic information as well. Genomics is going to be the study of the organism's genes, and this has provided new tools for classifying microorganisms. So because of their genetics, their genes, we can classify them and we can put them on a specific group in a phylogenetic tree, and we can also decide a specific treatment for that classification of microorganisms or just what diseases they cause. Recombinant DNA is DNA made from two different sources. So meaning, for example, you can put an animal DNA into a bacteria and make the bacteria produce that animal protein. Now, sometimes there's certain individuals that can produce a protein. For example, let's say a growth hormone or a certain protein. So what they do is they take the DNA of that specific protein or hormone, they input it in the bacteria, and since bacteria divide, very fast, now they're going to produce that specific protein or hormone, and then they extract it from the bacteria and then they inject it into the person. Okay, so it's just another way of producing something that not only humans but other organisms can produce fast. Biotechnology is the use of microbes for practical applications such as producing foods and also chemicals. So here we have the different dates, okay? You don't really have to know all of these. I know that it is a lot. The ones that you do have to know are Louis Pasteur for sure. Remember, he's the one that demonstrated that life did not arise spontaneously from non-living matter, but through biogenesis. Joseph Lister was the one that used phenol to pretty much apply aseptic condition, and this prevented a lot of surgical wound infections. We have Robert Koch, which he established experimental steps that link a specific microbe to a specific disease. We also can't forget Robert Hooke. He was the first one to observe cells, and Anton van Leeuwenhoek. He was the first one to observe animalcules or single-cell organisms. And then this one here, this is like the new modern, I guess, milestones for microbiology. You don't really have to know these either for your exam. But just so you know, here we have the first, I guess, the first person who discovered penicillin, which we know that it is an antibiotic. And then we have the specific genes that can cause cancer. We have even cell-mediated immunity, which we will learn in the last couple of chapters of the course. But just so you know, these are all still great contributions to microbiology, but you do not have to, I guess, know them for your exam. Okay, going back to one of the beneficial ways that bacteria do help us in a positive way is by recycling water specifically. Okay, so we know that, we have to recycle the water, we have to clean the water. So how it is done is by taking bacteria, and these bacteria, they like to eat the waste. Okay, they like to eat the waste. For example, they can convert carbon, oxygen, nitrogen, sulfur, and phosphorus molecules, which could be very harmful for us, and they convert it into something organic that can be used by plants and animals. Okay, so this is very, very, I guess, a great contribution to our water system, because we know that the sewage is 99.9% water, but only very few hundreds, which is like 1%, is suspended in solids, which is waste. Okay, so even though that it is only 1%, those are still very toxic, and of course, they're going to be very harmful for us. So the treatments to which is going to employ, like I said, bacteria, and they're going to remove undesirable components, just toxins, waste, from the water, and they're going to be then released and reused and just release something that is going to not be harmful for us. Okay, so bacteria, they do degrade and detoxify pollutants such as oil and mercury in the water. Okay, so as you can see, once again, when we think about bacteria, we say, okay, it's something gross, we don't like it, it causes disease, it's a microbe. But in this case, they do help us significantly. And you all have to drink water, so this is one way how they clean it. Microbial ecology is a study of the relationship between microorganisms and their environment. And this is only one relationship. There is other relationships from other microorganisms that, of course, affect us in a positive way, but they can also affect us in a negative way. And then going back to the pest control, we know that specifically the people that are in the agricultural field where they have crops, we know that insects, pests, they just eat their crops, and it's has affected them. This is how they live, and if they don't have good crops, then they can't sell them and make a living. So microbes are pathogenic to insects and are alternatives to chemical pesticides. So they did discover that microbes, bacteria, fungi, and other microbes, they do produce a toxin that is very harmful for this, insects, but it is not going to be harmful for the plant or even us humans that consume them. So what happens is here, for example, we have a bacteria, and here in this little dark area is the toxin. Now, when the bacteria is, disturbed, it is going to release the toxin, and then it is going to kill its target, in this case, the insects. So this is used by, to prevent insect damage to agricultural crops and disease transmission. So what they do is they spray the bacteria on the crops, and then every time an insect bites it off or just lands on it, they're just going to die. And this is also training the insects, okay, do not go there because we're gonna die. So a specific, I guess an example of a bacteria that produces this toxin is Bacillus thuringiensis. And this one is going to, produce a toxin that is very fatal for many insects, but it is going to be harmless for us animals and plants, which this makes it safe for the plant or the crop, and is going to make it safe for us to consume it. So like I said, the bacteria is going to produce these protein crystals, which look like this, and they're going to be toxic to the insects. Now, they even made an even more advancement on this specific topic, where instead of spraying the whole bacteria, they actually inject or inputted the gene that makes the toxin into the crop, and now the plants have that specific capability of producing the toxin. So technology, specifically in this case, is advanced so much that instead of, like I said, of spraying the whole bacteria, they just take the gene, and now the plant produces that toxin, which is very, very cool. Okay, so now, like I said, that we establish what microorganisms are, we established that they affect us in positive ways. We also said that they affect us in negative ways. And one of those negative ways is that they cause disease in humans, which is the most scary thing that they can harm us with. So we're going to talk about just several emergent infectious diseases. So what is an emergent infectious disease? It is a new disease and diseases increasing in incidence. An example, the very famous COVID-19. It was a new disease, and it just kept increasing and increasing over time. Now, when a pathogen invades a host or overcomes the host resistance, then the disease results. Okay, so sometimes, like I mentioned, the first line of defense from our body, it is not enough, and the disease or the microorganism starts winning in that aspect, and that's where you start to get sick, you start to have symptoms and all of that. So an example of an emergent disease is the Zika virus. This was first discovered in 1947 in Uganda through monkeys, so this was the first host. And then later it moved through humans, and it became a human epidemic because we made contact with those monkeys that were infected. In Micronesia in 2007, and then it even moved to the French Polynesia and Brazil in 2013 to 2015. So like I said, this was considered an emerging disease because it was a new disease that the cases just increase over time. So how this disease is spread is by the bite of an Aedes mosquito, which looks like this, and it can also be transmitted sexually. This tells you that it is transmitted through blood. Um, Now, when you, I guess, have this virus, you have the reddening of the eyes. You can even have fever, rash, and even joint pain. And this is because it does affect the blood. Um, the good thing is that the body can recover, but in the case of pregnant women, if you do get infected and you are pregnant by the specific microorganism, it can affect your baby and your baby can have certain birth defects. Okay, so that's why they are very careful with pregnant women, and they advise them to be careful with mosquitoes. Another one is the middle respiratory syndrome, MERS, and it is on the whole line of COVID or coronavirus. I'm sorry, coronavirus classifications. So this was caused by the Middle East Respiratory Syndrome coronavirus, or MERS-CoV. This one was not as severe as COVID-19, but it was one of the first ones to, I guess, affect people. Next, we have the common SARS, which is severe acute respiratory syndrome. This one, when it first started, it had 8,098 confirmed human cases, and it has 774 deaths in 2003. So this was considered an outbreak. Right now, it of course it's been detained, it's been maintained. But now we have the 2019 novel coronavirus, or 2019 nCoV. This was updated like at the beginning of the year, but in the US, there's been about 100 million confirmed cases and 664 million worldwide. In the US, there has been 1.08 million deaths and 6.7 million worldwide. Okay, of course, these numbers are high because, we're still getting people that have cases. But just so you know, COVID-19 coronavirus has always been there. It's just the different, I guess, classifications or species of the coronavirus that have affected us significantly. Thankfully, we are now in a post-COVID-19 era, which is great. But we still have to be careful with this specific coronavirus uh, classifications. And then we have also the H1N1 influenza. This one is very common. All of you know about this one. And it is also known as the swine flu. It was first detected in the United States in 2009, and then it later became a world pandemic, meaning affect many parts of the world, and a large-scale outbreak by the World Health Organization in 2009. So we're very familiar with the flu. If you are healthy, have a healthy immune system, your body can overcome it. But this one does target mainly elderly people or people that don't have their immune system as strong. We know that you can have fever, cough, headache, muscle pain, sore throat, runny nose, vomiting, and diarrhea. And like I said, there is vaccinations that are administered yearly, and this is of course to prevent people from getting infected and also having even a more severe outbreak. And then last, we have the methicillin-resistant Staphylococcus aureus, or MRSA. Now, this specific, I guess, bacteria or bacterial infection is very scary because it has been, I guess, becoming resistant to the treatments that have been used against it. Okay, so before it was, penicillin was used in the 1950s or before the 1950s, and then later the bacteria became resistant to it. So penicillin wasn't working. And then they came up with penicillin, which is just another antibiotic. And in the 1980s, this bacteria became resistant to it. And then in the 1990s, they then came up with vancomycin, which then they reported that it became resistant. So right now, I forgot the antibiotic that is used against this as of now, but I'm pretty sure that after the vancomycin, there has been several that have become resistant. But the Staphylococcus aureus affects mainly the skin. It is one of the common bacteria that is found on your skin. Normally, SRS doesn't affect you, but there are some strains that do eat off your skin. I know that this is very harsh, but this is an extreme case. As you can see, this person is missing almost all of her skin on this part of the face. It starts off like this, and you can treat it, of course, if you notice it. But back then there was a problem because the antibiotics that were used against it were not working, and many people were affected and even died because of this. So just something to think about of how scary and how microorganisms, even though they're very, very small, we can't see them with the unaided eye, how they can affect us significantly. This is the end of the lecture. Thank you.